Laurent Schwartz, a French oncologist, has been a prominent advocate for rethinking cancer as a metabolic disease rather than solely a genetic one. His work builds on the foundational research of Otto Warburg, who discovered that cancer cells exhibit a unique metabolic behavior known as the Warburg effect. This phenomenon describes how cancer cells preferentially rely on fermentation, anaerobic glycolysis, for energy production, even in the presence of sufficient oxygen, rather than using the more efficient oxidative phosphorylation pathway in mitochondria. Key points about Laurent Schwartz's work on cancer metabolism and fermentation. Cancer as a metabolic disease. Schwartz argues that cancer is fundamentally a metabolic disorder, where mitochondrial dysfunction forces cells to revert to fermentation for energy production. This metabolic shift allows cancer cells to survive and proliferate in low-oxygen environments, which is common in tumors. Lactic acid fermentation. Cancer cells convert glucose into lactate through fermentation, even when oxygen is available. This process is less efficient in terms of energy production but provides cancer cells with the building blocks, e.g., nucleotides, amino acids, needed for rapid growth and division. Schwartz emphasizes that this metabolic reprogramming is a hallmark of cancer and could be a key target for therapy. Targeting cancer metabolism. Schwartz and others have explored the use of repurposed drugs and metabolic inhibitors to disrupt the fermentation process in cancer cells. For example, drugs like methylene blue have been investigated for their ability to restore mitochondrial function and shift cancer cells back to oxidative phosphorylation, potentially slowing tumor growth. Criticism and challenges. While the metabolic theory of cancer is compelling, it is not universally accepted. Many oncologists believe that genetic mutations are the primary drivers of cancer, with metabolic changes being a secondary effect. Clinical trials targeting cancer metabolism, including Schwartz's work, are still in early stages, and more evidence is needed to validate these approaches. Fermentation and the tumor microenvironment. The fermentation process in cancer cells produces lactic acid, which acidifies the tumor microenvironment. This acidic environment can suppress immune cell activity and promote cancer cell invasion and metastasis. Schwartz's work highlights the importance of addressing the metabolic adaptations of cancer cells as part of a comprehensive treatment strategy. Practical implications. Schwartz's research suggests that targeting the metabolic vulnerabilities of cancer cells, such as their reliance on fermentation, could open new avenues for treatment. For example, methylene blue, investigated for its potential to restore mitochondrial function and inhibit glycolysis. Ketogenic diets, explored as a way to starve cancer cells of glucose, their primary fuel source. Repurposed drugs. Drugs like ivermectin and others are being studied for their potential to disrupt cancer metabolism. Conclusion. Laurent Schwartz's work on cancer metabolism and fermentation builds on Otto Warburg's discoveries and offers a promising alternative perspective on cancer treatment. By focusing on the metabolic adaptations of cancer cells, Schwartz aims to develop therapies that target the root cause of cancer's energy production. However, this approach is still experimental, and more research is needed to determine its efficacy and safety in clinical settings. Patients should always consult with their oncologists before considering any unconventional treatments.